All right, so today we're going to talk about a new project that we've been working on for a long time, and this is a phylogenetically aware package for phylogenomics. So it's a real tool that is a package that we hope that at least the Protist community might find interesting and will be able to use for developing phylogenomic data sets, but then not only that, but to develop or to further analyze those data sets whenever they're, once they have assembled those. So this is a project that's really come through, through several different paths. It was primarily through Martin and Laura in Andrew Rogers' lab, whenever we were all postdocing together. And we had decided that we were going to develop a tool, and it just never really happened. And then Fabian Berkey and Marek Elish had contacted me, and we sort of started to really develop that at a time whenever Tomasz Panik came to the lab. And so Tomasz had a Fulbright scholarship to come to the lab as a postdoc for about 10 months. And at that time, we were really interested in looking at heterolobosea deep phylogenetics. And we had used along with Alex Tice, had really looked at using the tools that we had already developed in the lab to address those questions with heterolobosea. But then we realized that the tools that we had weren't exactly adequate for asking those questions. And then we started to talk to David Giala at Marek's lab. And at that time, we had convinced him to come over and to do some programming for us. So this is the real team that has really has really developed the tool and has worked on the various aspects of not only the tool itself, but also the data set. So that includes Eric and Seraphim in Martin Kalisko's lab, who developed some of the computational tools also. And then along with, once we had really developed the tool, we had brought in Robert Jones, who is a fairly new PhD student in the lab, but is an unbelievable programmer. And so he has gone and cleaned up some of the code and has really made it ready for mainstream. So not only me and many of us on the Zoom meeting are very interested in very deep phylogenetics of the eukaryotic tree of life. And in order to do that, we really need phylogenomics. And so just so we can go back to what phylogenomics is this is taking multiple genes to infer a phylogeny, and then concatenating those all together to create a large data set matrix that we can then use phylogenetic tools to infer a phylogenetic tree. So what we have is, it often will be able to handle missing data, because in one taxon, we may be missing one gene, but we may have some other genes. And same thing for the next taxon. We may have some taxa that have all genes, and we can concatenate that all together. And in principle, this should be able to handle missing data from taxa. So one of the things that's very important when you're doing phylogenomics is the selection of orthologs and orthologous sequences in organisms is absolutely crucial. So there's several different caveats when looking at phylogenomics. Contamination that can be done, that can be caused by on-sequencer contamination, also endosymbiont contamination or prey organisms that the organisms may be eating. Along with that, there are also paralogs. So paralogs which are caused by genomic duplication, which may be either deep, such as like alpha and beta tubulin, or maybe within a group, so mid paralogs, or in paralogs, which are within a species or a genus. And we also are really interested in what are actually phylogenetically informative genes, and that also have a broad taxonomic sampling. One of the things that's very critical that we have found over the course of many, many years is that this also requires looking at trees and very careful consideration, so by eye. And that is a compounding factor that leads to, takes lots of time. And so we've also tried, we're still doing analyses by eye, just looking at trees, but we have some tools that will help with that. So phylogenomic matrices have evolved. And so the real major phylogenomic matrices, at least in protistology, or our history of Eric Baptiste and Hervé Philippe's data set, and then that has later morphed into Vladya's, and as well as mine and my lab's phylogenomic matrix. There's another data set that really was developed by Fabian Berkey and Jan Pawlowski's lab. And these data sets have more or less very similar orthologs. They have developed over time and morphed into various size data sets, and also can include various numbers of taxa over time. So not only does the data set has it evolved, we've also evolved methods. So the first methodology that Martin Kalisko and I developed when we were in Andrew Rogers' lab is a tool that we called OrthoLogger, and it's this phylogenomic pipeline. We never really published it and made it super widely available, though it was available to people if they contacted us. And I won't really go through that, but it has been used in lots and lots of publications and lots of, I would say, important publications. But PhyloFisher is a little bit different. So this is a new method, and it includes a lot of tools that allows for others to simply do phylogenomics. So it ships with the phylogenomic data set that will become available to everyone, and a large number of tools. It includes 305 taxa. This covers all of the deep eukaryotic groups. It includes 240 orthologs that we've whittled down from our Crumbs paper in 2018. And then we've fully coded this in Python 3, and we've made it extremely easy to install. So it can be installed using conda or pip with just a simple command. And also really importantly, this includes all dependencies. Those are automatically installed in sort of a conda or pip environment. So the PhyloFisher tool itself, so this is the Fisher algorithm that we have developed. And this algorithm was really developed through Alex and Tomasz. They really came up with the heart of this algorithm. And then that was coded by David. So when David was in my lab, he really wrote this program, and at least the initial part of the program. And Robert Jones has gone and cleaned this up a little bit. But what we have is two different types of methods. So we could have an organism that we don't know where it branches, such as Hemimastigophora, or we could have paramecium. So if we had a ciliate, we know pretty much where that's going to go in the phylogenomic tree of eukaryotes. And in our data set, which has 305 taxa, it also includes several ciliates, or quite a few ciliates. So if you have a paramecium, for example, that you wanted to add, you could say, well, we have some organisms in that database that can be used. So first we do a CD hit, and we cluster all the proteins from the proteomes that are predicted from the input data set. And then we do a hammer search, and those hammer searches are using profiles that are from the database of orthologs in that original database. We order those based on their significance, and then we ask the question, do we have specific queries of organisms that are closely related? So if we do, then we'll use BLASTP to take organisms from say Tetrahymena, which would be fairly closely related to paramecium, and we will blast those against our hits from our hammer search. And then we order those based on significance. But if you don't have that, then if you don't have organisms that are closely related, so let's say you don't know where Hemimastigophora goes, then you wouldn't have that, and you would just go ahead and collect all those hammerheads. Then we do a diamond blast against ortho-MCL, and we asked if if they're in the correct ortho group of ortho-MCL or if they're bacterial. And then we will do a blast against our entire data set to make sure that it goes within the right, the best hit is actually in the gene that we're expecting. So it's a reciprocal best blast hit. And then if we do have specific queries, then we are able to do a phylogenetically informed approach where we go and build a very fast tree and ask the question of, do those sequences actually cluster with our expected taxonomic group? If they do, then we'll order them accordingly. If they don't, then we'll still keep them. We aren't going to delete them, and we're going to put those into a data set. And then we'll have a data set. Then comes the very hard part, and that is tree inspection. And Eric from Martin Kalisko's lab was really instrumental, also with Tomasz and Alex, to look at all 240 orthologs and to examine these ortholog trees. However, they're not actually completely ortholog trees. Once we have collected n number of hits from various organisms, and we've done this from all 305 taxa, we've realized that we actually can keep the paralogy in these data sets. And then we're able to actually ask some questions about paralogy and include those paralogous sequences. What we realized very quickly is that looking at these trees is very difficult if you were going to look at them in fig tree. So we had Seraphim in Martin Kalisko's lab develop a tool. This tool allows us to input a tree that we open up, and then we can apply a TSV, which is just a tab separated value file that includes various details that are output from the Fisher algorithm, and that include some of that old metadata from the database. And this allows us to mark things such as paralogs. So what we can see here is that this is actually a paralog in this tree. I don't remember what tree we were looking at here. And then we have a bunch of sequences that have been added. And then we can go and look at each one of those sequences, but in a relatively fast manner. And we can mark what is paralogs. We know that this whole clade is a paralog. We mark those as paralogs. We may have some things that are contamination and may need to be deleted. These are pre-marked via the Fisher algorithm. And so we end up with a phylogenomic data set, and we have a phylogenomic tree here of the 240 orthologs. It's impossible to read, and that's fine. And I'll just show you this as a cartoon figure. And what we're seeing is that we've got some very interesting results. Primarily, the most interesting, I think, given Jana's talk last week in the ESOP program, is that we see TSAR with Hemimastigophora, and it's very highly supported, so at 99% bootstrap support. So we have PhyloFisher, which is a very easily installed and simple to use package. It ships with the data set. That data set, what's really, really critical is that data set also includes paralogs for each one of those ortholog alignments. So we have both a paralog data set and an ortholog data set, and this allows for us to have very accurate identification. It also will allow, and we've been testing this, is it will allow you to add your own gene sets, and these can be incorporated or used independently. And then we also have many tools for post-phylogenomic analysis. So often we see fast site deletion assays. We see random resampling of genes, which have become fairly common, where you're subsampling the data set and looking at the trees that are associated with those subsamples. But we also have amino acid composition clustering. We have a SR4 recoding. So this is looking at amino acid compositional bias. Also gene tree coalescence, species tree, which is input through Astral. We have a relative tree confidence score binning, which we used in Guy Leonard's paper from 2018 on hypochytrium, as well as identification of the most heterotachious sites. So this is a new tool that we are currently writing right now with Lori Eam and Andrew. And this, I think, is going to be really interesting. It's not completely done yet, but hopefully that will be done before we are out for beta testing. And then we also have a taxon collapser, which is ideal for single cell data sets. So if you were to go and sequence 9 different single cells of one organism that you think is one, and then you've gone and done single gene trees and you realize that they're actually or even a phylogenomic tree, you realize they're very closely related, you can actually collapse those into one single taxon. Also a tool for alternative genetic code prediction, and this is a pre-tool that you would use before you predict your proteomes. One single really quick word of caution and a word of warning is that over the course of doing all of these analyses and outputting alignments, we have a trimming tool that allows you to do prequel, MAF, GenZ, and then Divier. And this is a trimming.py. But we also have a trimming part, and that trimming part will allow you to use trimall or BMGE. And what I was really interested is looking at the effects of various trimming on the phylogenomic tree. So it was sort of a due diligence that we were trying to look at, because we are going to provide some suggested best practice for trimming, for example. And so for trimall, you can set a gap threshold. So a gap threshold of 1% means effectively no trimming, to a gap threshold of whatever you want, to 1, I guess. And that would be like 80% is our cutoff there. And then there's also very commonly used BMGE. So these obviously will, given various cutoffs for trimming, we see when you concatenate those supermatrices and using the tool Forge, those 240 orthologs will yield vastly different data set sizes. So what we have done in the past, and I think a lot of us have done this, is that we just infer a really quick tree through a site homogeneous model where there is one single empirical based amino acid substitution model for the entire matrix. So this entire matrix. And then we use that as a guide tree for a more robust model, such as a site heterogeneous model, which is very commonly used, like LGC60 PMSF, and PMSF is the posterior mean site frequency. So we have this guide tree that will give us, allow us to infer a site heterogeneous model, which allows for us to do some look at heterogeneous amino acid substitution processes among the various sites. So these are our classes. So the way that I have done this, and I'm definitely a person that has fallen for this, is that we have gone and we've done LG trees, which are computationally very easy to do. And then we use that into the PMSF model. But whenever I went and I looked at the various types, so gap thresholds of 1, 50, 80%, and BMGE, I saw vastly different topologies. And these topologies may be fully supported or 0% support. So for example, Hemimystigophoria plus Anchorocista is fully supported in the gap threshold of 1 and 0% support and gap threshold of 80. There are some clades that have no effects, such as SAR, Obozoa, and Crumbs, and Amoebozoa, but this was very scary. So we ended up then going back and doing a C20, so again, a C-series model, which takes a lot more time, 3 times the amount of time, to predict the site heterogeneous tree. And then we use that as our guide tree for PMSF, and it turns out that we yield the same results across the various trimming algorithms. So what we've decided to do is that we use the gap threshold of 80, which is sort of an in-between of gap threshold of 1 and BMGE. And then we can do further analyses, again, fast site removal, and this is what this tree is showing here, which is nearly a fully supported tree of eukaryotes. There are some clades that are contentious, but it's quite interesting, I think. So we have done alpha testing, that's pretty much completed. Now we're interested in finding beta testers. So if you're interested at all, please email me. And we're also really interested in engaging the interest of you all in a virtual workshop, which coronavirus makes this very easy for us. So now it's obvious to do a virtual workshop rather than meeting together. We were thinking in late July to mid-August of this year. That would likely be a 1 to 2 day program. And I would like to acknowledge, again, all of the co-authors, but particularly NSF and Fulbright, which have really made this possible. Without having Tomasz visit the lab, we would definitely not be at the point that we are. So with that, I'll take any questions, though I do need to get out of this so I can see you guys. Okay.
Great, thanks, Matt. I think you will probably have a lot of questions. Actually, the idea of a workshop sounds, at least to me, sounds very good. So I hope you can do it. So if you have a question, as always, just open your micro and go ahead and ask.
No questions?
Matt, can I ask? Can I?
Oh, absolutely. Absolutely.
Hey, Matt, thanks for the talk. Yeah, I'm good, thanks. I have a more technical question. I hope you forgive me on that. I was wondering, what is the tool doing? Let's say I have a long sequence which is cut in several chunks, but they all belong to the same autolog. So it's one sequence, but due to sequencing, it's cut in several chunks. Will it cherry pick the longest part, or will it include all of them and concatenate them and take them all into account?
That's an interesting question. So what we are doing, and maybe Alex or Eric, I think Alex, Eric, or Tomasz may be able to answer that slightly better. But we do have or actually Martin, but we do have a length cutoff that we basically, so it would collect all of them. Absolutely, it should collect all those sequences. The concatenation would probably have to be done by hand if you realize that those are within the same, the exact same protein. But we do have length cutoff filters, but the exact detail of that, I'm not entirely sure of.
Okay.
But yeah, that's definitely something that we have thought about.
All right. Yeah, because I just realized that, especially in the archaeoplastida, it happens many, many times that you have just sequence chunks. I don't know what's the reason for that, but yeah, when you don't go through it manually, then you would lose all of that.
Right.
Yeah.
Okay. Are there any other?
Yeah. Any other question to Matt?
You can just do proteins, right?
I'm sorry.
Protein genes. Can you find regulatory sequences and stuff like that? Because there's probably an awful lot of phylogenetic information hidden in that.
No, yeah, it's just protein.
Is there a way of expanding into the genome a little better, or a little bit more extensively, I guess?
As far as I know,
I'm just curious.
No, I mean, not that I'm sure there is. There's always a way. It's not something I've thought about.
Hi, Matt. I have a question. If you thought you knew what an organism was and didn't, or were wrong, and you put that into this, how would it look? Like, how would it tell you that you were wrong about your preconceived ideas about what it was and where it should go in the tree?
That is a good question that we have not, I don't think we've thought about. I don't
Just
know
to
how.
follow up, that you couldn't actually generate a phylogenomic support for your wrong idea that you would then put in a paper that looked like a right idea, would it? Like
I don't think
it wouldn't just give you a tree that said what you told it to say, would it?
I think that generally what you would see, so you're collecting multiple sequences from everything. And so even if it were your specific BLAST query were to go and reorder that, it's just reordering it. It's still going to be included in the actual single gene trees. And hopefully you would actually see that signal. We do definitely have a tool that provides you an overall signal within a tree and shows that there are some conflicting, like this is branching with high support lots and lots of times. That tool was fleshed out, but it ends up yielding like a massive PDF. And so right now we haven't completed that tool. I think Robert was working on that, but you would be able to see that there is a general consensus that is conflicting with what your preconceived idea was.
Okay.
Yes.
This is Andrew.
Oh, yeah.
Sorry, I'm done. There I am. Have you thought about putting other recoding schemes into PhyloFisher? Because the Ed Susko's program max-min chi-square will allow you to generate custom recoding schemes for your data to minimize compositional heterogeneity. So
this was Alex had brought this up probably a week and a half ago, and right, because SR4 may not be the best, and so we haven't yet. Okay. But we are planning on actually doing different schemes of recoding.
If you put the max-min chi-square program in there, it'll do it, and you can just stick them in, right? So.
Okay. Will you remind me of that?
Yes, I will.
Okay. Well, thank you all.
Yeah, okay, so any more questions, or we'll leave it here? So before you leave, Matt, so you mentioned that if people are interested in beta testing, contact you, right? Is that what you said?
That's correct, yep.
Okay, perfect. And how we will know about the workshop if it ever happens?
Well, if you contact me, even if you don't want a beta test, but you would be interested in the workshop, absolutely contact me. That's one of the reasons why I wanted you to record this, is I was going to put it on YouTube, and try to send it to the ESOP people. And hopefully it would be able to be more widely presented.
Great.
If you guys are okay with that.
Okay. Thanks a lot, Matt. Let's give Matt a big round of applause. So 3, 2, 1.